My name is Joshua Pan. I'm a graduate student at the program in genetics and genomics at Harvard Medical School. I'd like to present my thesis work on the functional analysis of protein complexes using genome-wide fitness screens. And the main biological question I try to think about is how do perturbations in protein interaction networks affect cellular fitness? And the main conceptual foundation I used to think about this problem was established by Lee Hartwell and Bruce Alberts back in the 90s. And they posited this model of the cell as this sort of layered functional hierarchy, where if you abstract away a cell's purpose as survival and cellular fitness, it encodes a set of cellular processes that respond to environmental threats and stimuli. These processes are themselves encoded by biomolecular machines or protein complexes that sort of govern the state changes that happen within the cell. But they themselves are composed of subunits that are encoded by genes. And so you can start to ask, if I introduce specific perturbations to genetic elements, and I precisely measure the fitness outcomes of those perturbations, perhaps across a variety of genetic contexts, could I learn something about the interaction network that's mediating that response from the perturbation all the way out to that phenotype? And I have a preliminary figure that sort of summarizes this concept for the audience here. And so to interpret this plot, we're looking at hierarchically clustered rows and columns. The rows are the genes that have been screened across a set of cell lines that are shown as columns on the bottom. And the height of each peak is the relative essentiality of that gene across that context. And the first thing that sort of strikes me about this plot is how variably essential genes actually are across these cancer cell line models. It's not just a flat line. And the other thing that sort of strikes me as well is that the way that this clustering has resolved these protein complex subunits as sort of these correlated modules of fitness effects across these cancer cell lines. And so it really suggests that maybe applying a network approach, we could actually start to curate and understand exactly which protein complexes encode this correlated fitness signature. Just a brief overview of the data sets that we've used. This is a deep collaboration with the Project Achilles group at the Broad. They both published these data sets very recently. One of them is a genome-scale RNAi library screened against 500 cancer cell line contexts and cleaned up with a statistical model that reduces the off-target effects of the RNAi constructs used. The second data set that we're using is this CRISPR-Cas9 genome-wide library that has been screened across 340 cancer cell line contexts and applied a similar model that actually reduces the copy number effect known as a systematic bias in the screening technology. And so the first question we tried to answer was a quantitative one. How many protein complexes that we know about actually have this significantly correlated fitness profile signature between its protein complex subunits? And to attack this problem, we used a network normalization strategy. And taking a correlation adjacency matrix, we would first do a row-ranked normalization. So take your gene of interest and take the top correlated gene all the way down to the bottom, keep those rankings. You then symmetrize that matrix by taking the top ranking between gene pairs. And then you can apply a network filtration approach, where you choose a threshold, and then you can create a set of edges between nodes, edges using only those correlations that exceed your threshold of interest. And so across these different thresholded networks, we then apply a simple statistical test. The test is based off of the hypothesis that we want to see an enrichment of internal edges versus external edges for our protein complex of interest. Namely, we'd like to see correlations that exceed this threshold between protein complex subunits rather than between subunits and other protein elements outside of the complex. We can then apply just a simple permutation approach where we take each edge and we rewire them while preserving node degree. And then in this permuted network, we can run that same, we can quantify the same ratio between internal and external edges. And that gives us a sense of that null distribution that we can then run an enrichment test on for our observed ratio. And so we applied this normalization approach across 5 different data sets that we wanted to consider. The first 2, which I've introduced already. The second is a co-expression database that is often used because mRNA co-abundance is often a measure of complex interactions. And finally, a set of recently published CRISPR screens that we're making the point here that the more screens you have, the better. And so what we're seeing here is across our different rank thresholds that we've chosen to filter our network on, we can take a cumulative recall percentage, and we're seeing that both the CRISPR and the RNAi data sets that I presented earlier are doing a fairly good job of capturing about 30% of the protein complexes that we know exist in a literature-validated set of complexes known as quorum. What's kind of fun about this is that there are a lot of protein complexes that are detected in both, which is great. There's a subset of protein complexes that actually appear only in the CRISPR-Cas9 data set. And it's interesting because those protein complexes are specifically depleted for annotated essential genes in the human genome. And that's suggesting that those essential genes are being depleted universally across these cancer cell lines. And they're not really giving us that variation profile that we need to use to infer these gene-gene relationships. Taking that all together, we built a curated set of 495 protein complex fitness similarity networks. And we then sort of proceeded to look at what these properties are of these networks and whether they matched up with our expectations for known solved structure protein complexes. And so the first one we decided to look at, and we did this systematically, but this is a great example, is the mediator complex. The mediator complex is a transcriptional activator. And it's actually a modular assembly of subcomplexes. Every large complex has to be built subunit by subunit, and they're often found in this modular organization. And so there's this head, middle, and tail modules that have been solved structurally recently. And if we look back at these sort of fitness landscape plots, we sort of observe that elements within the same structural submodule actually correlate with each other stronger than they do with elements outside of that submodule. And so we're looking at this again from a network approach here, using that same data set, just shown as a network. And we're seeing that these structural modules sort of have this community membership with each other, suggesting that we're actually kind of looking at a pretty deep level of this protein organization for that complex. We can perform that same analysis for the 26S proteasome. This is a structural interaction network built from PDB buried surface area interactions. And this complex is composed of the 19S lid and the 20S core. And we're actually seeing that in our RNAi correlations, these two structural modules are recapitulating as functional modules in our fitness similarity data sets. And so given this level of resolution, we decided to look at protein complexes that we knew a little bit less about. And the Kadosh lab that I'm a member of, we really study the BAF complex very deeply. This is mutated in over 20% of human cancers, but the actual function and structural organization of this complex is not that well understood. So we took that subunit list and we applied that same network filtration approach. And we found that there were these three correlated modules. And a graduate student in the lab, Brittany Michelle, ran an experiment where you can take the nuclear extract from a cell, you can run it out on a size sedimentation gradient, and then you can stain with specific antibodies to those subunits to see if there's actually a submodular organization as well. And what we found is that there's actually three strongly correlated modules that correspond exactly to these differently sized complexes, sort of revealing the modular organization of this complex. Lastly, one of the modules is actually a new one that no one's ever found before, and it's actually evolutionarily recent as well. We've been able to trace the exact duplication and divergence event that caused these paralogs to form a third subassembly. So it's suggesting that we're actually finding details of specific mammalian complexes that are of interest. To sort of close out our analysis, we decided to apply our framework on a set of unvalidated protein complexes to see what else there might be. And there's been a huge effort recently in the field to systematically map protein-protein interactions. And a group has done a really nice job of building an SVM model to actually predict more protein complexes than were detected in the original data alone by combining data from multiple papers and sources. But most of those are not validated. And so we took our approach and we used that CRISPR-Cas9 data set I've been talking about, and we ran the statistical test to see which one of their predicted complexes actually recapitulated as fitness modules in our data set. And we found that about 600 of them popped out as these really strong correlations. About 200 of them are known quorum protein complexes. And the rest, those subunits are in green up there. Some of them were recently validated in the literature, which is really exciting because it means that we're actually finding things that other groups are finding as well. We decided to focus on this blue module in the lower right-hand corner there. This module consisted of a set of tubulins. So there's an alpha, beta, gamma, delta, and epsilon tubulin superfamily. And the delta and epsilon tubulins are much less well understood than the classic other tubulins. We found that out of all of the proteins that are known to interact with the tubulins as annotated by these public data sets, really only two sort of popped out. And this was C16 or 59 and C14 or 80. We looked deeper into these two proteins, and we found that they actually correlated with each other as sort of two top-ranked global correlations in our data set. And what was really exciting is when we performed reciprocal IPs followed by mass spectrometry, we found that they actually co-precipitated each other, suggesting that they're a specific heterodimer. And finally, we confirmed the initial results that if you precipitate tubulin epsilon, you're pulling out these complex members as well. And finally, if you look at the subcellular localization of these uncharacterized proteins, they're sitting right on the centrosome, which is exactly what you would expect given the interactions with tubulin epsilon and delta. And finally, these two proteins are also recently evolutionarily present, about 400 million years old, around that same transition. So to summarize, we developed a statistical framework to sort of really test that first hypothesis, whether or not we could detect protein complex elements and predict protein interactions based on their correlated fitness profiles. And we're hoping to scale this up in the future to try to discover more than just protein complexes as well. And I'd like to thank my collaborators in this project, many members of the Broad Institute, my PI, Sigal Kadosh, and my co-lead on this project, Robin Myers. And I'll open up for questions. Thank you very much.